All right, hello. My name is Juan Manuel Vazquez, and today I'll be talking to you about the work we've been doing in establishing a clade of bats using chromosome-level genomes as well as cell lines to establish a resource for studying evolution of various traits, including longevity and longevity-associated traits. So my main focus is aging, and I think it's one of the most fundamental biological traits that we know of. Everything from bacteria to humans and all animals and all organisms age. And of course, one of the biggest problems that we are facing nowadays is aging in humans and all the aging-related diseases that go with it. So a lot of effort has gone into studying a variety of different species to study aging in humans, all these model organisms such as mice and how mice grow old, and dogs and how dogs grow old, and some other organisms. And so fundamentally what we've been doing then is understanding how short-lived species grow old and then the genetics underlying why they die and how they die in order to understand then the genetics governing short lifespans. However, there's a variety of different lifespans that we see in mammals. And so if we go beyond mammals to all vertebrates, is an even greater span of lifespans. So mice occupy this little niche right here of short-lived and small, but you also have things that are large and long-lived, such as humans, elephants, whales. And so if you really want to understand then how long-lived species get to be so long-lived, and more importantly, if you want to understand genes that can actually be used for interventions to extend human lifespan, what you really want to do is look at a long-lived species, something that's already very long-lived given its body size, and compare to other species that are closely related that live even longer, in order to understand then genetic interventions that may actually extend lifespan in those species, which may be better suited for extending lifespan in already long-lived species and extending healthspan, such as humans. So there's a variety of different animals that we can study, elephants, bats, and whales. But if you really want to hone down on understanding what makes lifespan and what causes changes in lifespan, you're going to want to remove all the big correlates. The biggest correlate is body size, and this was shown in 1991 by Austin and Fisher, that if you regress out the effect of body size on lifespan, bats become this extraordinary clade, where their longevity quotient, in other words, their lifespan residual outside after regressing out to body size, is a vertical line on this plot. So they have this non-allometric evolution of lifespan. In other words, an evolution of lifespan that's independent of body size. So genes associated with these longevity in these species doesn't make very large bats, just makes very long-lived bats. And so this means that bats may be actually a really good clade to study the genetics of longevity independent of everything else. And furthermore, in 2019, Wilkinson and Adams showed that there's another trait that for studying lifespan in these species that makes them very appealing. If you take all these longevity quotients and you plot them on the phylogeny of all these bat species, specifically the longest-lived Myotis mouse-eared bats, what you see is that the common ancestor was already quite long-lived, but especially in this clade of Nearctic Myotis, so North American and South American Myotis, you see that there's this increase and decrease in lifespan across the clade. In other words, in this short evolutionary time, 14 million years of this branch right here, you can see that there's actually this incredible dynamic range of changes in lifespan that has occurred. And these divisions right here between Myosocephalus and some other species sometimes is less than 1 million years, in which case you're talking about the difference between humans and other hominid species, but a doubling of lifespan in some cases. Furthermore, outside the context of the evolution of longevity, bats have a variety of different agonistically pleiotropic, as opposed to what we think of usually antagonistically pleiotropic, adaptations relevant to human health. Bats show an incredible ability to learn and long-term memory. They does not deteriorate with age. They have all these different adaptations related to flight, which increases their stress response, also associated with age. And of course, most famously, their bats and their strange immune systems and disease tolerance, which has been of special interest nowadays. All these things together means that by studying bats, you can get a variety of different insights that would not only benefit our knowledge of how different traits evolve, but also help us understand better how you can evolve tolerances to things that then can benefit human health. So I've been setting out for my postdoc to establish this system for studying the evolutionary genetics of longevity using functional genomics. And as I will talk about in this talk, we begin by actually generating these cell lines and genotypes from all these different bat species, and from cell lines establishing these chromosome level genomes, in order to then study them later with the cell lines to assess different functional traits. So these wing punches, you can go into the lab and establish these different cell culture tubes, and then go out into the field and collect these bats. If you all will just pinch the skin between your finger and your thumb, and that little flap of skin right there, just the edge, you'll feel no pain. And that's actually really convenient for the bats as well for us. That's the membrane that their wings are made out of. And they fly into cats, trees, cacti, all these different things. And so they're very used to having rips and tears in those wings and heal them very quickly. If you take a small wing punch biopsy from that wing, it'll heal up in a couple of days. And that same healing speed means that they grow in culture in a couple of days. So in a matter of weeks, you can actually go from out into the field collecting these samples to cell lines that have enough DNA to generate these ultra high molecular weight DNA for these different high chromosome level genomes. So for all the species here in green, we established chromosome level reference genomes, haplotype resolved chromosome level reference genomes using PacBio HiFi and Hi-C data. And shown here is the contiguity of the HiFi contigs relative to all the other previously sequenced bat genomes to date. And what you can see is that HiFi alone actually gives you this incredible contiguity that allows us to have the highest quality genomes that have been published to date. And then with the Hi-C phasing as well as scaffolding, we end up with chromosome level genomes with a countable number of gaps. Using these, I've been able to then take the cell lines and actually start doing some stress response assays to find genes that are associated with different differential aging-associated traits, such as DNA damage. And so what we've done is that we've taken multiple individuals of Myotis lucifugus and their cell lines, and we've treated them with etoposide, a DNA damaging agent, to then see what level of damage each individual has and how many die after a certain period of time, to find the LD of etoposide, and then use RNA-seq to interrelate genotype and phenotype. So we see a fourfold range in the LD50, in other words, a dose that kills 50% of the cells in culture for Myotis lucifugus, ranging all the way from this small nanomolar range to a micromolar and even millimolar range. So this shows that there's a very large amount of difference in between the species that may be underlied by the high heterozygosity of these individuals, which ranges in the whole species to about 1 to 2%. And these are the pathways that the genes that are differentially expressed are involved in, which, as you can see, there's some genes that we would expect, such as cell cycle and DNA damage. But there's also some genes such as that are involved in immune response, which may mean that there is some agonistic playtropy ongoing between their response to stress in terms of DNA damage, anti-cancer response, and some of their immune system genes that have been evolved in these different species. So with this system, we've been able to take advantage of the independent evolution and longevity in these species, to then establish these reference these reference genomes that are beyond human HG38 quality and approaching telomere to telomere quality. And using the cell lines that we generated in order to make these genomes, we've also been able to do functional assays that will allow us then to more cohesively tie in phenotype and genotype, and then more comprehensively functionally annotate the genomes and establish a robust methodology for studying evolution of traits such as longevity. In conclusion, I would like to acknowledge my home lab, the Sudemont Lab, and Peter Sudemont, shown here, for all of their help with this project. I would also really like to thank Rose Hadar, shown here, from Humboldt State, who has been indispensable for helping me generate all these cell lines. California Department of Fish and Wildlife, specifically Mike Bukowski, the director, and Devon Frazier, who was a postdoc in the CDFW, who helped me with lucifugus. And the NARD lab was indispensable as well for generating these cell lines, and Elise, who collected the developer individual, who then we've been making this genome with. And in my funding source, NSF PRFB, and I will gladly take any questions. Thank you.